Hello guys, welcome back to my EduSpace. Hope you all are doing and learning well. So today's video is in the continuation with the previous video of nucleus. Here we are going to discuss about the structure and function of nucleolus. So without further ado, let's get started. Before begin to our topic, let's take a look back what we have discussed till date. Okay, so we discuss about the shape of the nucleus, which is spherical in shape. The components of nucleus, which involves nuclear envelope, nucleoplasm, nuclear matrix, chromatin, and nucleolus. We have discussed about the nuclear pore complex, the nucleoplasm, the nuclear matrix in detail. So in today's video, we will make our understanding better about the nucleolus. Topics to be taken today. So we are going to know about what is the nucleolus, its background. Here we discuss about the experiments conducted for nucleolus, the nucleolus assembly at NORs, functions of nucleolus in rRNA synthesis and ribosomal subunit assembly. So you can see here in this diagram, this dark purple dot like structure, this is the nucleolus. Okay, and in today's video we are going to discuss about this nucleolus, how it forms, its structure and its function. So nucleolus, it is the largest component of nucleus found at the inside of nucleus. Okay, and it occupies spherical shape. So you can see this is our nucleus. Okay, and at the core of the nucleus, this dark stained structure, the dot like structure, is a nucleolus. Don't think it's a kind of structure, but it's just a domain, or we can say region, forming nucleolus. It looks like darkly stained dot in the heart of the nucleus. This one, okay? As it is not any kind of specific structure, so it is not considered as an organelle. Okay, this is not considered as an organelle. In fact, it is not membrane bound structure. Okay, so this is even not an organelle and nor even membrane bound structure. It is just a formation of some kind of domain or region which called nucleolus. It occupies 25% volume of a nucleus. Now, what are the constituents that made up this nucleolus? What are these components? Or we can say constituents. The nucleolus, this particular region is made up of rDNA, okay, ribosomal DNA, RNA and proteins. Nucleolus is nothing but an area where many rRNAs and proteins present. That I just told you that this is not any kind of structure, but it is just a domain where many ribosomal RNA and proteins are present. Okay, so you all know about the ribosomes. Ribosomes are made up of rRNA and ribosomal proteins. So this particular nucleolus region is carries synthesis of ribosomal rRNA, and it has nothing to do with the ribosomal protein formation. So nucleolus region can only produce rRNA, but it has nothing to do with production of ribosomal proteins. Now you must be thinking, if this particular region is only responsible for the synthesis of ribosomal rRNA, then why here assembly of ribosomal subunits takes place? So we will discuss this in our this particular video. Okay, so next, this is the background of Nucleolus. What are the history behind Nucleolus, its discovery and the experiments conducted? Okay, it is first discovered by Felix Fontana in 1781 and its name coined by Gabriel Gustave Valentin in 1839. The name Nucleolus was coined by Gabriel Gustave Valentin. Okay, John Gordon and Donald Brown in 1964 performed experiment on African clawed frog Xenopus levis for finding structure and function of nucleolus. Okay, they have performed an experiment on African clawed frog known as Xenopus levis for finding what is the structure and what are the functions of nucleolus. They concluded that those eggs which lack the nucleolus are not capable for life. So they found that 25% of eggs which are absent this nucleolus structure are not capable of carrying life. Okay. Max L. Bernstein et al. Found that DNA within nucleolus code for ribosomal rRNA. Okay, so this nucleolus, this particular region, the genes that belongs to this region is code for the synthesis of ribosomal rRNA. Now, what are the components of nucleolus? There are some regions inside the nucleolus where they perform some specific functions. So these regions are considered as the component of nucleolus. There are three major components of nucleolus. Like you can see over here, these are the three regions. The first one, second, and third. Okay, this particular area is our nucleolus. So the first one is fibrillar center, or we can say FC. At this location, transcription of rRNA from rDNA takes place. This particular region is responsible for the synthesis of ribosomal rRNA from rDNA and the process known as transcription. Okay. So here at the fibrillar center, transcription of rRNA from rDNA takes place. Now the second is dense fibrillar component or DFC. After this rRNA synthesis, okay, at this location, pre-rRNA processing takes place. So when the rRNA is formed here at the DFC region, the processing of rRNA takes place. Now the third region is granular component or GC. Here assembly of ribosomal subunits takes place. At this region, here the ribosomal rRNA, okay, rRNA combines with the ribosomal subunits. Ribosomal subunits. Okay. So here assembly of ribosomal subunits takes place. Next. So now the assembly of nucleolus. You all must be curious that if nucleolus is not any kind of prominent structure, so how it forms? Basically, it is the junction of a specific part of chromosome, that is ribosomal DNA, which codes for rRNA synthesis. So you can see in this picture, here we have this chromosome. Okay, I'm taking just one. This chromosome, that particular region, this one, that particular region of chromosome, or we can say the DNA strand, is concentrated at the center. We have such 10 chromosomes. Okay, so this particular region is our DNA, which codes for the synthesis of ribosomal rRNA. So the nucleolus structure is formed around a specific area of DNA known as rDNA. So this is our chromosome, and this particular area which contains that specific genes which codes for ribosomal RNA synthesis is known as rDNA. The rDNA is arranged in tandem repeats. Let me draw this. Suppose this is our rDNA. Okay. Now let me understand you what is these tandem repeats. So we have same sequence of genes which are repeated again and again. Okay. Which codes for rRNA synthesis. So these small stretches of genes which grouped together, they form rDNA, and these are the tandem repeats. Such rDNA region is known as nucleolar organizer region, or NOR. So we can say this particular region, or this particular region of ribosomal DNA is known as nucleolar organizer region. So its name suggested that this particular region is responsible for the formation of nucleolus. So it is known as nucleolar organizer region. So we have such 10 chromosomes that are having this NORs. But it doesn't mean that if you have 10 nucleolus, okay, this is not true that each NOR is responsible for the formation of a nucleolus. Even 10 such junctions of NORs are responsible for the formation of this nucleolus in human being. So in fact, multi-NORs participate in the formation of a nucleolus in an eukaryotic cell. So let me tell you some more about what is this NOR, that is nucleolar organizer region. Suppose we have this much chromosome, okay? We have this chromosome. This is what? This is our centromere. Okay, this is centromere. This is centromere, and suppose this stretch of DNA, This particular stretch of DNA is our NOR. This one. Okay. Now, in this nuclear organizer region, we have grouping of genes which are arranged in this fashion. Okay. The same sequence of genes are repeated again and again, which are going to transcribed to our RNA from our DNA. So, such type of chromosomes, their numbers in our cells are 10. So, we have 10 such type of chromosomes that are having the NOR region. And we also know when this, I just told you that these chromosomes are junction at the center. 10 such kind of chromosomes are junction at the center and form the structure known as nucleolus. How many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this particular region is our nucleolus, which is formed by the junction of such type of 10 NORs. Okay. So this particular part is our rDNA. This is our rDNA, which is responsible for the synthesis of ribosomal RNA. So if we magnify this particular gene, we will see that these are the RNA coding sequences having 18S, 5.8S and 25S. These are the coding sequences which are interrupted by these small ITS regions. Okay, which are not coding for any sequences. So these are spacers, ITS spacers, and these three sequences are repeated again and again. So these are tandem repeats. Here we have the promoter gene. This promoter gene is responsible for the initiation of transcription process. Okay. Next. Now the synthesis of ribosomal rRNA. Okay. How the synthesis of ribosomal rRNA from rDNA takes place. We know transcription means formation of RNA from DNA. So here transcription process takes place where rDNA is responsible for the formation of ribosomal rRNA. Suppose this is our nucleolus, okay, and this is the DNA strand. I am taking just one, but such 10 NORs are responsible for the formation of ribosomal RNA. So this is the DNA strand, and those stretch of DNA that is present inside this nucleolus region is our DNA. Okay, having tandem repeats of gene sequences. These are the tandem repeats of genes. Now here, from these tandem repeats, transcription process takes place. Okay, and the formation of ribosomal rRNA. Okay, so the tandem repeats of rDNA is responsible for the synthesis of ribosomal RNA through transcription process. The whole region where nucleolus is creating ribosomal RNA from rDNA is known as NOR. So this particular region is known as NOR. Okay. We know that for a ribosome to form, we need RNA. We need RNA as well as what? We need RNA plus ribosomal proteins. Okay. And ribosomal proteins. But we just saw that this particular area is just responsible for the synthesis of ribosomal rRNA, but from where we get this ribosomal proteins. So for this, we have to go to outside the nucleolus, or we can say outside the nucleus. This is nucleus. This is our nucleus. And these are nuclear pores. So we have to just get out from the nucleus to the cytoplasm to get our ribosomal proteins. This is cytoplasm. Okay. This is cytoplasm. So inside the cytoplasm, the ribosomes are constructing ribosomal proteins. These proteins has to be transported to the nucleus to the NOR through nuclear pore. Suppose this is our cytoplasm. Here we know that there are so many free ribosomes are present. These free ribosomes are responsible for the formation. They are constructing ribosomal proteins. So these ribosomal proteins are then taken inside the nucleus through these nuclear pore. Now these ribosomal proteins comes in this nucleolar region. Okay, this is our ribosomal protein. Now we have both constructing units of ribosome. These are rRNA as well as ribosomal protein. So now the assembly of ribosomal subunits. These ribosomal proteins then join with their respective rRNA and finally form ribosomal subunits. So inside the nucleus or nucleolus, the rRNA, rRNA are going to join to their respective ribosomal subunits. They are joined with their ribosomal proteins to form ribosomal proteins to form ribosomal subunits. Now they are going to form ribosomal subunits. Ribosomal subunits are small subunits and large subunits. Okay, so these subunits once again leave the nucleus through nuclear pore and enters to the cytoplasm where the large subunits join with the small subunits to form complete ribosomes. So here the ribosomal subunits are formed. Some are large and some are small. But here the large subunit is not going to assemble with the small subunit. For this, they have to again go to the cytoplasm. Okay. Here the small subunit is going to assemble with the large subunit. So now the formation of ribosomes takes place outside the nucleolus region, that is in the cytoplasm, where the small subunits, that is 40S, is going to attach with the large subunit. Okay, this is large subunit and this is small subunit. In our case, the small subunit is 40S, whereas large subunit is 60S. Now the formation of ribosomes, as I just told you, once they join and form full ribosomes, they can initiate protein synthesis as soon as mRNA, that is messenger RNA strands, get attached to the ribosome. So in the cytoplasm, the ribosomal units are assembled, and once the mRNA get they are going to synthesize what this they are start synthesizing proteins, okay, by the process known as translation. So translation always occurs in the cytoplasm. Hope you understand what is the nucleolus and how rRNA synthesis takes place inside the nucleolus region, and how the ribosomal proteins are get transported from the cytoplasm to the nucleolus, and how they get assembled the respective ribosomal RNAs to their respective ribosomal proteins and form the subunits of ribosomes. And now these ribosome subunits get transported back to the cytoplasm, where the synthesis of proteins takes place, and the small subunits get assembled with the respective large subunits of the ribosomes, and they form the full ribosomes. Okay. Now the functions. Now the functions of nucleolus. So we have discussed that the main function of the nucleolus is to the formation of ribosomal rRNA, or the synthesis of rRNA from rDNA. Here the processing of ribosomal rRNA is also takes place. Here the assembly of ribosomal subunits also takes place, and it can detect cellular stress. This is a new discovery where the nucleolus is responsible to detect the cellular stress. Okay. Thanks for watching.